Hello, hi everybody. So, dear students and friends, today we'll be learning about substances which are very essential for our daily life. Maybe I can say without these substances, there will be no life at all, I guess. So, those are your enzymes. So, that is the substances or the proteins which are synthesized in our body, and yeah, they are helping us to lead our life. So let us start. What are enzymes? We'll learn about these enzymes, how they are, what are their nature, what are the functions. So let's start. So we all know for any chemical reactions we need a catalyst, right? So the chemical reaction is practically possible only with the help of catalysts, right? So the catalysts are the one or the substances which accelerates the rate of chemical reaction. So that is how these enzymes are the catalysts. They are biocatalysts. So they will uh, accelerate the reactions and they catalyze the biochemical reactions in our body. So let us see how the catalysis takes place and how they perform their catalytic reactions. Okay. So first we'll see what are the objectives comes under this topic, enzymes. That is how you define the enzymes and what are all the terms we use under this topic, that is active site, apoenzyme, holoenzyme, prosthetic group, enzyme specificity, and the structure of the enzymes, how they act, I mean to say mechanism of action, classification of the enzymes, how they are classified, and what are the factors affect your enzyme action even in vivo and in vitro. Okay, so let us start our journey. So first we'll see what are all the features of your enzymes. What is the nature of the enzyme? What are all the features? So first thing, the all your enzymes, whatever are present in our body, they are all proteins. They are made up of the amino acids. Okay, they are like proteins. They are protein in nature. And these enzymes follow the same physical and chemical reactions of proteins because they are protein in nature, and they are heat labile. They cannot withstand in heat. They are not heat stable. They are heat labile, and they are water soluble. You can dissolve, I mean they are water soluble. Okay, and they can be precipitated by any of the protein precipitating agent, whichever we use to precipitate the proteins in our media or in our solution, in our mixture of the solutions, I mean any fluid. You can precipitate them by using a precipitating agent, by adding a precipitating agent. So example I have given here are trichloroacetic acid, or you can use the ammonium sulfate. Okay, so that is precipitating agent, and there are about the 60% of weight of your enzyme is nitrogen. So we all know the proteins have their amino, they are made up of amino acids, so obviously the nitrogen will be there. So the 60% weight as a nitrogen. Coming to the importance of enzymes. Okay, what are all their importance, or what role they will play in our daily life. Okay, and other than that, so the enzymes mainly play an important role in the metabolism, that is your metabolic reactions, whatever takes place in our body. Okay, all the biochemical reactions are enzyme catalyzed reactions in all the living systems. It may be a plant or a mammal or a human, whatever it is. So the reactions is catalyzed by the enzymes. And these enzymes can also be used as a therapeutics, means we can use them as a drugs, or you can use them for treatment of some of the diseases. For example, enzyme can be used, as I said, they can be used therapeutically, such as digestive enzymes. For I have given the example pepsin. Pepsin is a digestive enzyme. We all know the pepsin, which is present in the, which is a proteolytic enzyme, so that could be used. And along pepsin and trypsin also could be used for digestive disorders. Okay. And streptokinase, yes, that could be used for as a one of the therapeutic drug or therapeutic enzyme. Okay. And coming to the diagnosis, so there are many enzymes which are used to diagnose some of the disease. Okay. Levels of the enzyme in blood, serums, or plasma, or any body fluids can be used as a diagnostic important enzyme, or for the diagnosis of any of the disease. For example, if I take CKMB, that is your creatinine kinase enzyme, which could be used, or which is a very good indicator in myocardial infarction. So that is an enzyme. Okay, so that is how you can use the other example, AST. ALT stands for alanine transaminase, which could be used in any of the liver disorders. The levels may shoot up in your blood sample. So that is how these are the very much important things of your, importance of your enzymes. So coming to definition, how you will define them, how you define these enzymes. Enzymes are defined as biocatalysts because they are the substance which will catalyze the biochemical reactions in our living system, right? And these are the proteins that increase the rate of reaction by lowering something called activation energy. Okay, they will lower the activation energy. We'll see what is activation energy in the further slides. And the enzyme catalysis is very rapid. For example, I can say it's very rapid, means one molecule of an enzyme can act on 1,000 molecules of any of the substrate, its specific substrate, per minute. Means in 1 minute about 1,000 substrates can be converted to products by the enzymes within a minute. So that's how the rapid reaction it is, and their reaction, and they are not altered or consumed during this reaction, and they are reusable as they are not consumed or not altered. So you can use them, reuse them, or our body reuse that enzyme. So lack of enzymes will lead to any of the inborn. For example, if there is any enzyme defect in the particular pathway, metabolic pathway, example I can take phenylalanine hydroxylase is an enzyme which comes in your phenylalanine amino acid metabolic pathway. Okay, so if this enzyme is deficient or lack of this enzyme in our body or in this metabolic pathway, leads to something called as phenylketonuria. It is an inborn error of metabolism of phenylalanine, the amino acid. And another example, a very good example I can give is my favorite, lactose intolerance is a disease. It's an inborn error of the enzyme defect lactase. Okay, so there will be a deficiency of lactase, leads to a disease called lactose intolerance. So this is how there are many different enzymes. Deficiency leads to many different inborn errors in our body. Okay, so this is about some simple introduction of your enzymes and its definition. So let us go the terms what we use in this topic. First thing is active site. So the active site is the region of the enzyme where substrate goes and binds and where the catalysis actually takes place, right? So the region of the enzyme where the substrate binding and catalysis occurs is called as active site. So the active site contains substrate binding site and even a catalytic site. Sometimes these two may be separate. Okay, so that is the active site. So let us see. So here you can see the active site. Enzyme molecules contain a special pocket or a cleft is called as the active site. Here you can see the half moon shaped with the depression so the half moon shape or the cleft with the cleft is your enzyme and the depression is your active site. The depression is your active site and the green color is a substrate, okay, which is going to bind on that enzyme, I mean to say in the active site, and then after binding it forms something called as enzyme substrate complex. Once the complex is formed, it goes to transition state and then the actual catalysis takes place and then the products are released. So that is how the enzyme the substrate goes to bind with your active site. So this picture what it is showing is your 1 minute. I'm trying to pull this pen. So here sorry for so this is a substrate and here you can see this is a cleft which is showing the active site. Okay, so then when the binding is done, so that is called the enzyme substrate complex formation, and here it goes to the actual catalysis and then the products are formed which are released now. So this enzyme is now free, then you can again utilize for the next reaction. Okay, so this is about the active site. So active site is nothing but the region on your enzyme where the substrate goes and binds. Okay, so coming to the substrate. What is substrate? So this term is a substance upon which an enzyme starts its action, on which the enzyme starts its action is nothing but your substrate. The reactant in biochemical reaction, okay, is a substrate. When a substrate binds to an enzyme, it forms an enzyme substrate complex, as I said. So this is your substrate here going to bind here, this is your active site, and that forms a enzyme substrate complex. And as I said, where the enzyme actually starts its action is called as, or the substance is called as substrate. Okay, so the substance upon which an enzyme starts or acts is called as substrate. Okay, so next is mode of action of enzymes. How the enzymes are going to act on their substrates. Okay, so we have some theories to explain the mechanism of action of enzymes, and there is something called lowering of activation energy. So in previous slide we heard about activation energy, right? So how it lowers, or what is this activation energy, first we'll see. So activation energy is defined as the energy required to convert all molecules of reacting substance from the ground substance to transition state. So you are supposed to convert all these molecules to their transition state so that there will be actual reaction takes place and they are converted to the product. For example, it is like when you are trying to make a fire, okay, even if you keep on lighting or keep a flame, you don't, I mean, you cannot burn initially. The wood will not start, I mean, the wood will not start burning. Why? Because you need some initial supplement or activation energy for them. So what you do, you will put some paper or some kerosene so that they start initial burning, then the whole wood starts burning, right? So that is how, similarly, the activation energy is something which is initially supplied, okay, to be initially supplied. So this will be reduced by your enzymes. So enzymes reduce the magnitude of this activation energy. Okay. Example, I can say, if you hydrolyze the sucrose manually, okay, you need around 26,000 calories per mole of your sucrose. Okay. For the activation energy without enzyme for the hydrolysis of sucrose require 26,000 calories per mole, okay, while the action or activation energy is only 9,000 calories per mole when you will hydrolyze the same amount of sucrose by the enzyme called sucrase. So it is 26,000 when it is done without enzyme, that is by using a acid hydrolysis, that is 26,000 calories per mole. If you use the sucrase enzyme for the hydrolysis of your sucrose, then you need only the 9,000 calories per mole. So there where we are using maybe 3 to 4 times more energy when we use, or more calories when we use the acid hydrolysis of sucrose. So this is how the activation energy will be reduced when it is by the enzyme. Okay, so that's how enzymes will reduce the magnitude of this activation energy. So coming to the theories, as I said, we have some theories to explain the mechanism of enzyme action. The first theory is Michaelis-Menten theory. So this theory is put forward by these two, Michaelis and Menten, in 1913. This is called the enzyme substrate complex theory. According to this theory, what they say is E plus S, which gives ES complex plus E plus product. So here the E, what it's showing is E is nothing but your enzyme, S goes to substrate, and P for product. ES is nothing but your enzyme substrate complex. So these are the so enzyme binds with your, substrate goes and binds to the active site of your enzyme. Then there is a formation of enzyme substrate complex, and then the reaction takes place. After catalytic reaction, the enzyme is free now. It releases the product, and then the free enzyme is available for further action. So this is the Michaelis-Menten theory. So coming to the next model, that is your Fischer's, that is your Fischer's template theory, or the lock and key model. So this states, so this theory or this model states that the 3D structure of the active site of the enzyme is complementary to the substrate, like your key is complementary to your lock, right? So that is how this the structure here in this model, only in this model, this 3D structure of the active site will be a complementary to the substrate. That is how it explains us, this theory explains us about our enzyme and substrate, or the active site and the substrate. So thus enzyme and substrate fit each other here. Okay, so here you can see the picture is showing where the appropriate key is available for this. So here this is the key, I mean to say substrate, which is going to fix properly into this active site, so that this is the correct. Okay, and we have a wrong key, you can see here this is the wrong key, so there is no proper reaction. So that means this model is very rigid and it is not flexible. Okay, so it's like a complementary key for the lock, so that is why it is called as lock and key model. And this is another picture which is showing the same. So here you can see the active site shape is exactly how the substrate, I mean it is complementary to its substrate. So if it is exactly complementary, then only it can form a proper I mean binding with the your enzyme. Okay, so as per this model, the enzyme structure, as I said, it is rigid and it fails to explain many facts of the enzyme action. Okay, and the allosteric activity of the enzyme action, like allosteric modulation, for example, you cannot explain the I mean modifiers action, mean to say inhibition and all, you cannot explain with this model. So that is the drawback of this model. And coming to the other model, which is widely accepted model, that is Koshland's induced fit theory, or Koshland's induced model. This is put forth by Koshland in 1963, and here the conformational changes are occurring at the active site of an enzyme. You can see here, so this is your substrate with this shape, and here you can see it's not complementary to your substrate, right? So here you can see the cleft is little different, I mean the shape is not exactly what your substrate is having. Okay, the structure. So after binding of the substrate, there will be a conformational changes is occurring in your active site of an enzyme. So this, so the substrate here is inducing some conformational changes in the enzyme after it binds to your active site. So here this model is not rigid, it is very flexible, it is flexible, so that after binding only it is going to I mean there will be a changes of conformational changes takes place. You can see, you can give an example of wearing a gloves. So it fits to your fingers, so that is your Koshland's model. That is how they are flexible. I mean the structure of this model is flexible, and it helps us, and it can be used to explain the action of allosteric modulators. You can explain the competitive inhibition also, whereas you cannot explain this competitive inhibition by using the previous model, which is Fisher's model, that is lock and key model. Okay, so that's how this Koshland's model is widely accepted model for your enzyme action. So in the next session we'll learn about the classification of enzymes. By this I'm ending the simple introduction of your enzymes, its importance, and some simple terms like your active site, activation energy, substrate, and then the different models, theories, that is your Michaelis-Menten theory, and the Koshland's induced fit model, and uh, the Fisher's template theory, or the lock and key model. So thanks for your patience. So let us see the classification of enzymes in the next class. Till then, have a good day. Bye.